Hello everyone, I'm Alejandro Montenegro and I'm Executive Editor for Cold Spring Harbor Protocols. I'm joined today by Philip Benfey, who is a professor at Duke University and A2TMI investigator, to talk a little bit about the research and the work that we'll present at the 87th Cold Spring Harbor Symposium, which this year focuses on stem cells. Thank you very much for taking the time.
Sure.
So research in your lab is focused on specific plant cells derived from meristems acquire their identities. Tell us a little bit about these meristems and their location and function.
Sure. Well, my interest in plants really came from the simplicity of the system. I was not trained as a plant biologist. I was trained as an immunologist, my PhD. But I also, my deep interests were, how do you get this incredible multicellular entity that we are, or the plants are, from single cells? So that's from the stem cell perspective, this was quite germane. And what I saw in plants, and particularly what we work on mostly, is in the root. You have a stem cell center at the tip of the root. And from that stem cell center, and it's just 4 populations of cells tightly packed to each other, all the other cells in the root are generated from that. Moreover, because the cells don't move in relationship to each other, that stem cell generates its next cell up, and that cell will then divide to make whatever the progenitors of various tissues. But each cell, each new cell that is made is added to the bottom of that column. And then what it means is that on the longitudinal axis, you have a developmental timeline. Everything in a single root, you have everything from the least differentiated cell to the most differentiated cell for all the different cell types. And that's an enormous simplification compared to what happens with a mouse embryo, where you have to look at day by day, et cetera. So that's really what attracted me to the system. And we've been working on it for 30 some years now. And there's still new things that are popping up every day.
So you mentioned that your work has primarily focused on the root. Why focus on the root versus other stem cell niches?
Yeah, the root has the additional simplifying aspect that it's, at least for Arabidopsis, it's translucent. So we can put it under a microscope and we see all those cells. We can see them divide. That's what I'll be showing in some movies that I have for my talk. And that has greatly aided our understanding. Moreover, the root, as compared to the shoot has a stem cell niche also, but the stem cell niche in the shoot works on a more probabilistic basis. Cells divide, and then which becomes what cell type is much more probabilistic. In the root, it's all, at least of Arabidopsis, cell divides, it becomes that, whatever that lineage is going to be. So you have a complete and total lineage definition just by looking at those different cells.
But in the root, even if there are external cues that wouldn't affect the specific determination of the lineage?
So the main thing that would have an effect is damage. So if you damage one of those cells, it's very interesting. What happens then is that a neighboring cell will divide, will push into that area, divide, and then that cell that's come into that space will take on the identity of the cell that it's. And so it's a completely, in developmental biology parlance, it's a completely regulative system. Even though you have these lineages, and it looks like those lineages should be the things that are defining everything, it's really where the cells are, their neighbors, that are defining who they are and what they become.
So in your talk, you will highlight the use of single-cell transcriptomics to identify gene regulatory networks that function within different cell types and developmental stages. I was wondering if you could tell us a little bit about the insights that you found in this context.
Sure. So single-cell transcriptomics has the great beauty that we can now, if we take a root, dissociate the cells, we can put it back together. We can, from each cell, actually in our case, we knew so much about what was expressed in different cell types that instead of doing the normal clustering, which is what you normally see with single-cell RNA-seq, we could assign an identity to every cell, and we could assign its relationship to the other cells by how closely, how much of an overlap there was in the transcriptome.
The trajectory.
And so from that, you get the full trajectories. Stem cell, we get everything from the stem cell to the fully differentiated cells and the different tissue types, et cetera. And one thing that we seen here, and we can't be 100% sure this isn't some sort of artifact of the system, but it definitely corroborates other data that we have, is that you rarely see genes that are turning on at a very sharp point and then turning off at a very sharp point, which you would expect if it was a series of switches.
Right.
Rather, you see things turning on and then they sort of fade out. Something else will turn on, fade out, turn on, fade out. And we think that one way of explaining that is that differentiation is not so much a series of switches, but rather is a probabilistic process. You get a bunch of genes, the right genes, and then it has a high probability of becoming cell type X or cell type Y. But it's not this will happen because this gene is on in this cell.
Mm-hmm. So you can manipulate the what genes come on or off and
absolutely
change the fate?
Yep.
Mm-hmm.
Yeah.
Mm-hmm. So you have also leveraged high-resolution time-lapse imaging to study differentiation in the root and to identify also transcriptional regulators involved. What have you found using this approach? How has it complemented the transcriptomic data?
Sure. So we, based on essentially transcriptomic data, but that we got originally based on just isolating different lineages, et cetera, as well as genetics, et cetera. We and our colleagues, Ben Sharas and others, came up with a model 10 years ago that what was happening in the root, there were two regulators that we had identified, short root and scarecrow, and these regulators were key to making a very essential asymmetric cell division in the single cell. So the stem cell divides, gives its daughter divides longitudinally, and then those two cells will go off and make two very different lineages. And all the data at that time, we had all this data, and we collaborated with mathematical modelers. They used ordinary differential equation models, and they came up with something that was very satisfying. It made a bistable switch. Mm-hmm. So it was bistability, emergent property, and we thought, well, that's good. And probably, if we were a good biologist, we'd just say, okay, we're done. But we decided that we really wanted to see how this happened, not just, yes, there was some sort of switch. And when I say bistable switch, it meant that at a low level of expression, you got nothing happening. High level expression, that's when the asymmetric division occurred. And there was very little that happened in between. So there was sort of unstable situation. And so we, primarily a postdoc in my lab, Kara Winter, set up a system using a light sheet microscope where you could watch the root grow in real time and grow over 48 hours. So, and tracking the tip of the root, and then labeled these two proteins, the short root and scarecrow, and watched as transcriptionally induced short root in a short root mutant background, and watched it so you could see in time. You could see how much there was there. You could see it actually physically moves to the next layer over. You could see it inducing scarecrow, and then you could see the asymmetric divisions. We also had a normalization factor in the nuclei of these roots, and with that, you could actually quantify how much protein there really is at each time. And the results did not fit the bistable model at all. There wasn't a high or low level. You had various different levels before the asymmetric division. Moreover, very strikingly, a very small amount early on of short root expression would still result in an asymmetric division 5 hours, 10 hours later. Mm-hmm. And that did not fit the model that we had. So we were a bit stuck at that point. And we were fortunate that another postdoc, Pablo Skelly, joined the lab, and he had a background not only in computational biology, but also had worked in industry for a while and done a fair amount of machine learning work. And he said, well, what if we just take a machine learning approach and just say, what are all the features that could influence what's going on? And it turned out that the feature that was most prominent was related to the cell cycle. So it turns out that, and so all of our data now point to the fact that you don't need a high level of short root, you don't need a high level of scarecrow, you need just enough at the right time. It makes sense. There are a lot of things just in time, but that wasn't what we were before. Now we have a very different picture. So if you express just enough of these proteins early in the cell cycle, you get the division to occur. If you don't get enough, or if you can make them as much as you like later in the cell cycle. And it won't, you have a sort of 50-50 chance at that point of getting an asymmetric division.
So what's next then in this program?
Yeah, so what we would love to know now is, what is it that actually causes a division? This is very striking. You have symmetric divisions and these asymmetric divisions. What is it that causes that shift in orientation? What are the genes that are involved in that process? And how does that physically happen? And we now think we have a system where we can induce these asymmetric divisions pretty much, not exactly at the same time, but almost at the same time. On the other hand, we can inhibit them, so we get almost asymmetric divisions. And we have approaches both from microscopy, time-lapse, single-cell RNA sequencing, that we think we can identify the components and then watch in real time as these components act in the cells and see how they actually make these shifts.
All right, so that's all the time we have today. So thank you very much for taking the time.
It's a pleasure.
And thank you all for watching.